So our first speaker today is Jochen Roeper, who is from Goethe University and is going to tell us about pacemaker mechanisms and plasticity of dopamine substantia nigra neurons. Take it away.
Okay, thanks. So let me just get ready. Okay, thanks, Veronica. I hope you can all hear me. It's a great pleasure and honor to be selected, to be also talking dopamine in a virtual fashion. I've never done that before, so it's great fun. Hello, everyone out there, friends, colleagues, and lab members. Thanks for tuning in. So I'll be talking about pacemaker mechanisms and the plasticity of that pacemaker in situations where we lesion part of the dopaminergic system, so it has a broad relevant background to Parkinson's disease. Let me kick off by using my 20 minutes to focus on two excellent, very talented MD PhD students, Joseph Shin and Laura Kovacheva, who within the pandemic had all the time in the world to finish their beautiful projects. And this is, I'm going to tell you everything that's unpublished now, but very ready to go out soon. So I'm focusing on them, but of course, there's lots of other people in the group who have contributed, and my dear collaborators help you out there, and I'll mention you as I go along. So let me take you on a drive. Our dopamine neurons tick away, whether we are awake primates or anesthetized rodents, about five times a second. That's 15 billion in a lifetime. Not all make it, about 10%, 4% die every decade, even if you don't get neurological disease. So this is a long drive, and there's a big discussion whether that drive is too long. We have to protect these guys because they're very precious. But in a more computational sense, most people are interested what happens when this baseline activity of 5 Hertz in this narrow bandwidth is violated by interaction of the circuit, because that's when the fun and the function starts. So the dopamine cells, as we all know from the Schultzian paradigms and the modern instantiations by the Ishida lab, by Lama lab, and many others here in the conference, that when for a short time they fire very fast, what's called a burst, then we can signal things like positive reward prediction errors. When they pause for a while, they can communicate very important signals. And we heard many talks about this already, which might, among others, act as reward prediction, negative reward prediction errors. So if this is a car, then driving along the road is only one job. We have to go into these different countries, which I'm, as a cellular electrophysiologist, would say that the pause country and the burst country. But I will focus, and this is a bit unusual probably, on staying on the road. What does it take? What's the machinery, and how is that changed? And as you will see, there are a number of, maybe a surprising number of open questions, although for the last 30 years, people have really looked deeply into that machinery. A new approach with my friend Carlos. Hi, Carlos, you're out there from San Antonio. So we joined forces 5 years ago to make it possible to do in vivo patch clamp recording of dopamine cells, which has the big advantage that you can look below the hood. You can look at sub-threshold activity, which is where all the action is. So how action potentials are generated, what pattern, what frequency, is mainly decided before the spike occurs. And it's basically about the integration of synaptic input and the role of intrinsic channels. And most of you know that even if you rip out the dopamine cells out of the brain, make them isolated cells or slice cells with synaptic inputs inhibited, they still, like the SA node in the heart, beat away. They don't beat quite at 5 Hertz, maybe just half the frequency, but a major contribution is that they have an inbuilt motor called the pacemaker to generate that rhythm. And this slide just reminds me that compared to the awake primate, which was a Paul Glimcher paper, very beautiful paper, quantifying the spiking properties, in a mouse it's almost the same. So we fire around mean frequency around 5 Hertz, the CV, which is a measure of the variability, how good can I stay on the road, how much do I go to both sides, is also around 40%. Yeah, so very similar, highly conserved, one might want to argue. So the cool thing about in vivo patches, now we can see, as I said, what's going on in the subthreshold range. And one of the insights that Carlos and I got from the first study, this is anesthetized, so there's no behavior here to talk about, is that when the in vivo dopamine cells pace in this narrow bandwidth, that in the subthreshold space, the variation of the minimum potential and the threshold potential is very, very small. You see in B and C from that 2020 paper how precise and unimodal that distribution is. When that unimodal distribution changes, the neurons can escape and create other interesting patterns like burst or pauses. I won't be talking about that today. So we can reformulate the question in a more mechanistical sense to say, how does a dopamine cell in vivo and in vitro manage to keep this precise control on subthreshold voltage range, given that it's bombarded by up to 8,000 synapses? So how does it manage? And it's not kicked around like a Poisson process. So this is where biophysics and channel interest really starts to become relevant. Mainly the beautiful work by the Bruce Bean lab, this is a classic landmark paper from 2007, Roeper et al. Have shown that, and this is isolated cells in vitro with no synaptic input, that in the interspike interval range here, which is the one that controls when the next spike will occur, is dominated. They're very small currents, but it's dominated by a cobalt sensitive, that is a voltage gated calcium current. And pharmacology from that lab and many other labs clearly demonstrated that there is a role for dihydropyridine sensitive, also called L-type calcium channels. And they come in two variants, one low threshold classical pacemaker channel that's also very important in the heart, it's CAF 1.3, and a high threshold, more ubiquitously expressed channel called CAF 1.2. So even with the best drugs available, you can't distinguish between these two. And I will come back to that, how to solve that. So Joseph, who summarized the literature of the last couple of years, you see like the last 30 years, plots here dihydropyridine concentrations used and the effect it has. And all you need to see, it's diverse, yeah? This is usually my territory. I would say, oh yeah, this is because of dopamine diversity, but that's not a trick that will always work. Sometimes it's just like, well, what is the relevant concentration of the drug you have to use? In clinical terms, and millions of people taking drugs for high blood pressure that will have low nanomolar concentration of dihydropyridines in their brain, maybe we'll come to that, already affecting dopamine cells. So this is the clinical range. You see that most of the studies in vitro were in the micromolar range and had heterogeneous effects. Only few recent studies, including the Somaya lab and the Liz lab, were looking into a low nanomolar range that really occurs in vivo in humans treated. And they didn't see any effects here. So that's the situation where we, together with Jörg Striesnik, also collaborate and say, what's really going on? What's the problem? What's the function here? So Joseph combined a number of in vitro approaches, on-cell recording, perforated recording, dynamic clamp, wide dynamic clamp, so you can extend the firing range. That's only just a few hertz in vitro across the entire dynamic range that you observe in vivo. So you can boost it up to get up to 50 hertz, which wouldn't occur spontaneously, because we were interested in how that channel would operate in the entire range that dopamine neurons can populate. And importantly, at least the beginning of the study, we used a knock-in model where only the CAF 1.3 channel, the pacemaker channel, the low threshold channel is sensitive to the drug. It's been mutated out of the other channels. So we can be really precise about the molecular target here. So that's a nice trick. And this is a model designed by Jörg Striesnik's lab in Innsbruck. So we did very careful perforated patch recordings because those aficionados like Rebecca know exactly that all these channels are highly modulated and they wash out and they change things and you might miss something. So Joseph did that, very carefully controlled, and then he washed in a saturating but still selective dose of isradipine, a dihydropyridine that's actually used in humans. And what he saw is there's a significant decrease of frequency. But as these two examples say, it depends. It depends on the starting frequency. So it's a frequency dependent effect. If the cell is active only around 1.5, 1.8 hertz, which often occurs in slices, there is no effect. If it paces faster, there is a clear effect. And other parameters are really not changed. So the variability is not changed. So you can plot this, and what you see is that that depending on the starting frequency of the cell, there's a linear amplification or contribution by Kaf1.3 channels, which is about 30%. So if you have a cell pacing at 5 Hertz, about a third of that is mediated by the activity of Kaf1.3 channels. So it's an amplifier. It's not necessary for basic rhythm. It's not the magic channel that we're all trying to hunt down, the one that starts dopamine. And Vincent Soitin and I are working on a project like that, but it's like an amplifier, but it's frequency dependent. So that's number one. Now, of course, we want to work on wild-type mice, so where we pre-incubate different concentrations of isradipine. Now that we have a linear correlation between the block of the channel depending on the starting frequency, we can make predictions. So what Joseph did in the DHP 1.2 mutant mouse and in the wild type, he looked at controls, then he said, okay, what will be, how would this population change if the linear amplifying is correct? And he predicted the distribution that he would experimentally observe using isradipine. And as you see, in all 3 instances, different genotypes and different recording conditions, the prediction was very precise. So we think we can generalize and say that indeed in substantia nigra neurons, Kaf1.3 pacemaker gain is also operative, right? Now he wanted to extend still in vitro into the range which really is populated by dopamine cells in vivo. We use, again, in collaboration with Carlos Palladini lab, who pioneered this in dopamine cells, we use the dynamic clamp. So in silico, we inject NMDA conductance to push the cells into much higher frequencies. And you see different cells that this gets quite a broad range. If we sorted them by principal component analysis into different components, we'll see that the high frequency responders, the upper range, are all populated in the lateral part and the low responders here. So we segregate the population. And of course, that's very resonant in my lab, where we study differences of lateral and medial nigra, and I might come to that. So now we looked at only the lateral cells, which have a very high response. And again, over the entire dynamic range, the predicted contribution of Kaf1.3 is completely happening when you block the Kaf1.3 channel. So there's a good match between 1.3 channel function, but across the entire range. So that simplifies, I think, a functional attribution of that channel. Says 1.3 just amplifies by 30% whatever the cell is doing, even if it's firing at 50 Hertz. This is only true for lateral cells. I don't have all the data to show you because of time reasons that that's not true in medial cells. Let me just point to that, the reason why. In medial cells, when you get medial nigra cells, if you get to high frequencies, another, probably an N-type channel is opening, and that calcium is used to couple to SK channels as a negative feedback. So only lateral cells are linearly amplified, while medial cells and ViDA cells, they basically use a high frequency negative feedback with other voltage-gated channels, just as to say that. The next and the final step is to say, okay, fine, that's what it does. What's the situation in vivo? We have to change one important thing. We have to go down to the clinical range. The clinical range is not 300, but 10, maybe around 10 nanomolar. So he titrated that concentration in vitro and showed that now we're only blocking partially 1.3 channels, that the linear correlation goes down with the slope. So it's now what we are affecting is something like a quarter, 25% of the frequency is reduced. So that's the prediction we have when we go in vivo. And here, and these in vivo experiments done by Laura Kovacheva, because they both teamed up in both projects. So we record a single cell, which then later will be labeled, so we know exactly that's in the lateral nigra, and it's dopaminergic, of course. And then we systemically inject 3 mg per kg isradipine, which is close to the human dose, and we observe what's happening in these cells. And what we see here is that the baseline activity of these lateral cells goes down. We look at the distribution. we'll see that it goes down in a way that is very close to what we predicted from in vitro. So it's a very simple situation here that nothing else seems to matter. We can replicate the in vitro situation in vivo, and we get a very similar slope. So cells that fire away at 8 hertz will take about 2 hertz away from their firing. So if we plot this, it actually also predicts a concentration that is reached around these cells in vivo. And together with local pharmacologist, Professor Geislinger, we determined the concentration, and it is very much in the similar range. So we think that at least for lateral, and other data shows that they are dorsolateral stratum projecting dopamine cells, that we can affect them with clinically relevant doses. The implications we can discuss, but I think that's a starting point, at least for the mouse. Okay, let me change gears for the next kind of 10 minutes. And of course, this is just wild.
Jochen, sorry, we have 5 minutes left.
Yeah, 5 minutes, sorry, okay. So I'll speed up if I can, go from 5 hertz to 8 hertz now. So this is Laura's work, and we wanted to know, how does the pacemaker change when we are in a post-lesion situation? This is not a Parkinson's model, which is progressive. This is a 6-hydroxy partial striatal lesion, taking out about 50% and then see what happens. Behaviorally, we see that there's a motor phenotype, a turning bias that then completely recovers over 60 days. So Laura looked in vivo at the early stage, and she found a massive but selective deficiency in the surviving cells, mostly in the medial nigra, because in the lateral they were all dead, of bursting. So we actually really, in a factor of 10-fold, lose selectively the bursting. If we do in vitro, and this is Joseph's work at that early point, when also behavior is impaired, we see that the pacemaker isn't altered, but it is becoming irregular. So it is unstable, as if it's going to depolarization block, recovering. We all know this when we have cells that are not really very healthy. So maybe this is an early damage state, but the interesting thing I want to stress is what happens after 2 months. So behaviorally, at least the rotation bias is gone. We don't see any sprouting. We don't see any more dopamine cells. So we think it must be at least a functional contribution. And what Laura saw is that now the ACSF injected and 6-hydroxy injected are the same. So one might argue, speculate that they had the time to recover. The question is it pure homeostasis? Do they just come back from damage to be normal dopamine cells? Well, now, and this is the point I'm getting at, and it's the final data, but no, they find a new set point. So now if we record in vitro after that 2-month period, look at the survivors again in the medial nigra, we'll see that the pacemaker has doubled. So they have radically, they have significantly changed the set point of pacing at twice the speed. We don't see that in vivo. It's probably balanced synaptically, but in vitro, the core machinery is really altered, and that's highly significant. And the final point is, what are the channels that control the frequencies? Well, I showed you something about the gain, the CAF 1.3 channel. Here we see that one of the big breaks, the A-type KB4.3 channel, is actually responsible for that phenotype. We can occlude this pharmacologically, as you see here, and final data slide. And if we look at the protein expression semi-quantitatively, we see that it is clearly downregulated on the affected side. So in essence, I took you through a drive to dopamine cells. I showed you the cockpit, and let's look at the cockpit again. We identified for one population a simple accelerator, the CAF 1.3 channel. I showed you, and that was known before by work from many labs, including our own, that CAF 1.3 is an important break, but that break is altered, partially not really working when we have surviving cells. And I didn't talk about how to change gears, how to go into different modes, which is another really fascinating topic. And with that, and hopefully roughly in time, Veronica, I'll stop talking. I'm happy for questions. Thanks a lot.
Excellent. No, actually, you really speed up, and now you have 1.5 minutes left.
Oh my God. Okay.
Oh, it's good. It's good, I think, because there are questions already popping in that I'm going to let Paulina take. But if I may ask actually a question first myself, I wanted, while more questions keep coming, I wanted to ask you on how, very interesting on the first section of your talk, when you described the differences and the important role as I like it, as an enhancer or accelerator of the calcium channels, on how you think this work of yours lab relates to Jim Sarmeyer's work, where he proposed that the expression of these channels was actually also leading in part to the damage. So, or like create these more lateral cells to be more vulnerable to then death. So I just wanted to hear your take on that.
Yeah. I think Jim's idea is great. I'm a subscriber and a fan. I think the evidence was a bit problematic because these channels didn't, as I showed you initially when we started, didn't really increase the activity. We didn't know whether they work in vivo. What we did know, which is very important, is that they were responsible for dendritic calcium oscillations. But we don't really understand what the dendritic calcium oscillations are doing on top of controlling the pacemaker. So I think our study might contribute to simplify the picture, that in particular, the most vulnerable substantia nigra neurons, the dorsolateral projecting area, actually uses that channel every day, in a mouse at least. That in principle, we have the concentration range is in people, and you know, and that you're referring to this work that people who take isradipine for antihypertensive reasons have a 30% reduced risk of Parkinson's. And they take it when all the cells, we assume, are intact. Now, the clinical study that didn't show a protective response only starts after clinical diagnosis. And we know from pathology, the beautiful Cordover and Halliday work in Brain 2013, that even a year after the initial clinical diagnosis, there's almost none of those cells left. So that's why I think it's important. The second part, what are the cells we can protect? Well, can only protect the survivors. So we have to find much earlier biomarker. I think these channels maybe have a potential for a isradipine, do you have a lateral nigra left clinical test? So I think that's an interesting development that I would love to see human physiologists and clinicians taking over. But also to really closely monitor, do we reach a concentration? And that's a debate, and I'm not an expert on this, but do we really reach the concentration that we can affect the activity? And remember, final comment, this is acute. Mm-hmm. What is happening to this machinery if we put an osmotic mini pump and then treat them for a month? Do they upregulate the channel? Do they try to escape? Do we cannot block them for long? I think there's a number of really interesting open questions, but coming back to the key point, I think Jim Sarmeyer has a great point. And I think even if that first study failed, I think we really should reinvestigate and focus more specifically on those cells to see whether there's a chance to protect them. Definitely.
Excellent. Thank you. Pauline. No.
Thank you. So we have a couple of questions. So the first one comes from Rebecca, and she's asking, does the capacitance change in the recovery dopamine neurons, or do you see any increase in their size?
We have not looked very carefully, but the obvious sort of somatic proximal dendritic capacitance, it did not change. And many of the other parameters that we looked at in vitro and in vivo have not changed. So mostly when I didn't talk about it, they have not changed. There's nothing else. We don't see a hidden factor. We think the adapted cells might have also a little bit of an HCN upregulation, but it's probably subtle compared to the KB4 effect. Yeah.
Okay, so another question comes from Paul Kramer, and he is asking, DANs also laterally inhibit each other through dendritic dopamine release and activation of D2 GERK inhibition. Did you look at all at GERK inhibition in the ablated region?
So, hi Paul, great question as usual. So there's another project by Niklas Hammer, who works on projection-specific identified pairs of dopaminergic cells and asks the question, who's ruling, who's inhibiting who? So it's an entirely different topic. We have not looked at this in this project, but what we found in a nutshell is that there is some within projection inhibition, via dopamine synapses. But the biggest effect is that lateral cells inhibit limbic projecting cells. So that seems to be in our hands with testing a number of projections, a clearly hierarchical communication within the nigra. Very different topic. What would be cool, and we haven't done the combination, is to ask, is the local communication when one, for instance, the lateral cell is dropping out, is that rewiring? Is that plastic? And these are possible questions now, but we haven't looked at it now.
Okay, perhaps we have time for one or more questions. So we have one from David Salser who says, Jochen, congratulations on a nice finding on the control of pacemaking. A question becoming, does a stimulus that would drive bursting have a different bursting response if tonic firing is reduced?
Right.
Mm-hmm.
Hi, Dave. Great question. Do I know the answer? Well, I know that what's fascinating, and we don't fully understand in the compensated chronic state, that bursting has somehow managed to recover. That if we do, and I didn't show you the data, if we do the dynamic clamp and just inject in silico NMDA, which is not the full story, of course, because there's no calcium coming in, we see a similar response range. So we have so far no mechanistic indication to say how burst responses might be altered. But of course, there are caveats and limits of our study, and Dave knows exactly what they are. These animals are not awake, so we don't know what the dopamine tone is. We don't know whether there is long range or short range feedback. All that hasn't been addressed, and I think should be addressed before I have a convincing answer today.
Perhaps one more?
Good, yeah.
Okay, so we have a question from Amir Kashif, and he's asking, is there any relationship among the dopamine neurons' individual activity, for example, synchrony?
You mean a cross correlation between pairs of dopamine cells?
That's what I would infer, yes.
Yeah, okay, Amir, thanks. Great question. We haven't looked at this in this study. So we have another study by a junior PI, Cyril Duvaci, published with multi-electrode in vivo recording. And there we had about 60 or 80 pairs of triplets. And similar to the work in the monkey, there wasn't much under baseline conditions of a synchrony or coherence. But in that study, which has a very different background, whenever the animals started to work in a working memory paradigm, then the cells became partially entrained in a 4 Hertz rhythm, and then there was much more synchrony, very similar to what the Bujaki lab has shown in the rat a couple of years earlier. So not in that it is happening, but in that context here, we don't know. And I think it also relates to the other question, what is the role and what's the changed role of dopamine-dopamine interaction? Does it synchronize? Does it desynchronize? What's really going on in wild time? What's going on in that situation? These are all open questions and I think cool questions.
Excellent. Thank you so much, Jochen. That was a great talk and discussion.